person that is presenting is Robin Owen. They are a professor in the Department of Biology at Mount Royal University in Alberta. The title of their talk is Reconciling Estimates of Diploid Male Frequencies in Populations of Euglossine Bees, Allozymes, and Microsatellites.
I know I've sent to see everything. Sorry about that. I'm not terribly technologically advanced. Anyway, good afternoon, everyone, or good morning or good evening, depending where you are in the ether right now. But today I'm going to talk about diploid male frequencies in euglossine bees, these very colorful orchid bees found in South America. And of course, diploid males are produced generally by inbreeding in populations, and we're not concerned about the actual mechanisms, though often there's a single locus sex determination system, but they are potential indicators of population decline. Ambrosiad and Lawrence Packer have shown that diploid male extinction vortexes can occur under situations, and therefore they're very important for conservation. Now, obviously, when we're estimating something that often is in small proportions, it's critical to make the best and proper estimates. And actually, a paper by me and Lawrence Packer was published in 1994, and this did set out all the theory required for using marker loci to do these estimates. However, this paper has often been not understood or ignored by various authors, and so there's a complete variation, a mishmash of ways in which the proportion of diploid males in a population are reported. So it's very difficult to compare populations and species, but also there's clearly a lack of basic understanding by some authors on how to do the estimation. So in this talk today, I will review and clarify the theory for estimating the proportion of diploid males in populations of Hymenoptera using multiple microsatellite loci. Then I'll reanalyze data from papers both with allozymes and microsatellites, and this will allow a comparison across species and populations in the standard format, and it will actually lead to considerable reconciliation between the divergent estimates of diploid male frequencies in these bees. Now, microsatellites, of course, have been used extensively for the last few years, but it does appear from reading the literature that the people who use microsatellites, who are referred to as microsatellite people, tend to look down on those using allozymes, because after all, microsatellites are looking at DNA itself. They use sophisticated techniques, PCR and so on and so forth. They have many different variable loci at their disposal, whereas the allozyme people are still mucking around with chemicals, making up arcane recipes to look at obscure enzymes, and therefore, it's a more primitive technique, and therefore thought not to be quite as good. And in fact, some of the microsatellite people have criticized earlier allozyme studies on a number of different grounds. One is that of low heterozygosities. The other is to say that allozyme-based techniques are really not very good, and there can be considerable misscoring of alleles. Now, this, I think, is a case of people who are living in glass houses throwing stones, because as we'll see, the statistical techniques used by the microsatellite people aren't that reliable, and this second criticism does seem to be rather ad hoc and ad hominem, not based on any real evidence. However, they're obviously saying this for some reason, and there is a real discrepancy between allozyme and microsatellite studies. Now, the paper I'm using as a sort of a whipping boy, if you like, for the microsatellite work is this one published in Evolution by Souza et al. In 2010. And I will present a bit of a reanalysis of some of their data and simply point out where their their misunderstanding has come in. Now, as I pointed out, there is a considerable discrepancy in many studies between microsatellite estimates and allozyme estimates. Interestingly, the microsatellite estimates are rather low, often around 0, whereas the allozyme estimates tend to be rather high, even up to 0.9 or 1 in some cases. But also notice the interesting inverse relationship between height of investigator and size of estimate. This, of course, is completely spurious and will not be discussed further. But when I was looking at the various papers and the data, something just didn't seem quite right. And there must be some middle ground really in reality. And when I look at things like this, I always like to think of the quotation by Richard Feynman, the great physicist, and the top one I particularly like. And this I interpret as, if anybody claims to be absolutely right about something, almost by definition, they must be wrong. And certainly the bottom quote is very true, that science is not certain. So what I did was to go through the papers and reanalyze the data and put it in a common format. But what I'm going to go through next briefly is the whole idea of using marker loci from and looking for diploid males, and lead you through it quite quickly, but it's very, very easy actually. And you'll see precisely how we should estimate these diploid males and pinpoint exactly where the microsatellite people have become misled. So if we have a population with haploids and diploids in, both diploid males and diploid females, we can look at the proportion of diploid males as either the proportion of diploids that are male, or as the proportion of males that are diploid. And it's this second one, phi, small phi, is the most operationally useful one, because mostly, and particularly with the orchid bees, where it's easy to sample lots of males, you have a sample of males and you want to estimate this phi. So we're sampling from a population, and this is simple binomial sampling. And of course, if one were able to completely identify every single male as haploid or diploid, there would be no problem at all. So if you did flow cytometry or looked at the chromosomes of each and every male, then you would have this sampling and you wouldn't have to go any further. And of course, it has the normal binomial variance. But the problem is, of course, that that's not usually practical, and we use marker loci. But first of all, I should point out that when you're doing binomial sampling, you do have to obviously have a decent sample size. Now, as we'll see, many of the microsatellite people have used sample sizes of 10 or less, even 3 or 4 or 1 or 2 ridiculously. And of course, the smaller the sample size, for realistic values of phi, the probability of getting more than or even a single diploid male is very low, as you can see here. Now, normally, in most usual populations, you would expect the frequency of diploid males to be about 1 or 2%, maybe up to 5%. So as you can see, having a decent sample size obviously is important. So if we use marker loci now, then the problem is, of course, you can only detect diploid males if they're heterozygous at that locus, because haploids and homozygotes of diploids get mixed in together. So the estimate, and this was originally published by Packer and Owen in 1990, and then fully developed with the sampling theory in Owen and Packer 1994, is this basic estimate. And now the variance, of course, is not binomial. It takes a little bit more working out because you've also got the allele frequencies in there. But it's very obvious. It's the number of heterozygotes observed divided by basically the heterozygosity times the total number of males. Now, if we successively rewrite this equation and add alleles and loci, you'll see what we end up with. So equation 2 can simply be written like this. Equation 3, we're separating out the binomial sampling from the heterozygosity. We can add multiple alleles very easily. We can extend it to 2 loci, as Owen and Packer did. And then it's easy to extend to multiple loci, L, with many alleles at each one. And as you can see, the basic form of the equation is the same, but it's very, very obvious what's going on. And if we look at equation 6, we've got, I'll just pop on to the next one quickly and then back to it. We've got in one set of brackets, the binomial sampling. The other is essentially the heterozygosity. And this is what is termed the probability of detecting a diploid male with multiple microsatellite loci. And the trouble is, this p-det is, in fact, what has been used generally by the macrosatellite people. They've ignored the other part of the equation. So there's a few things that follow. Of course, as the number of marker loci increase, equation 6 produces to equation 1. We've become back full circle to the initial starting point. But also, once p-det reaches a certain level, about 0.99, then nothing more is to be gained by adding more marker loci. And as you can see from the table here, once you get to 3 or 4 loci with about 100 males, that's perfectly adequate to detect any diploid males if they're there, and you've got the adequate binomial sampling. And you see that adding loci really doesn't change anything. You're changing the sample size very little, and also tweaking to the 3rd or 4th decimal place of the p-det is simply unnecessary. Now, just briefly, I don't expect you to read all these tables, but I reanalyzed the data, put it in this common format, starting with the Sousa paper, and you can see the number of populations that need to be eliminated because, somewhat arbitrarily, but I think sensibly, n of 10 is simply the minimal you can use. So number of populations highlighted in yellow simply have to be eliminated because of the very small number of males sampled. And then what I did was to take all the other papers on microsatellites and recalculate everything in terms of phi. And as you can see, there's quite a range. And these are some of the most recent and more comprehensive papers. But we still now have quite reasonable phis in many cases. Certainly many populations don't have any diploid males, others do. Then I did the same thing with the allozymes. And as you can see here, that basic Rubik paper that started this debate does have fairly large phis, but when you go on to the other papers, again, there is quite a full range of estimates, including zeros. And when you finally compare the number of populations that are examined by allozymes and those by microsatellites, there is much more consistency than was originally thought. And as you can see, there still is more microsatellite percentage of populations with low diploid male frequencies, and the ones with high diploid male frequencies are generally the allozymes ones, but there is a considerable range of overlap getting up to fairly high frequencies that seem to be entirely consistent between both methods. So in conclusion, it is clear that diploid males are reasonably common, at least in some populations of euglossine bees, but we need to look at population by population basis rather than species as a whole blanket statements such as that the diploid males are very rare in euglossine bees, or very common, are simply not informative. It is best to have comparative estimates, for instance, this phi, which can be used over methods, allozymes, or microsatellites, populations, and species. And we must get our sums right. Otherwise, any recommendations we make from this, of course, are meaningless. And if we do think it's important for conservation, this is really critical to get this done correctly. And those are the references. I've given the link to this picture, which was actually from the Frost Report, a British comedy show in 1966 with John Cleese and the two Ronnies. I thought that was a rather nice way of comparing the way that microsatellite people look at allozyme people. Anyway, that's all I need to say today. I don't really have any acknowledgements, and this was an unfunded research and so on and so forth. So thank you for your attention. And if there's time, I can answer some questions.
So there isn't any time for questions, unfortunately. So we'll just move on to the next speaker. But like everyone else, just if you guys have questions for Robin, just send them to him via the DMs.
Okay, thank you.
So yeah, so the next person who is presenting is Jason.